Pharmaceutical Processing, in conjunction with INTERPHEX, presents INTERPHEX Live.
Flexibility as a cultural constraint within a firm. I mean, what do you see there? What's the understanding out there? And yeah, right Ed, just turn it on and just. You got it? It's on.
Is it on? Okay. So the when it comes from what flexibility, so if you think about pharmaceutical manufacturing, the cultural constraints are what most people have grown up in the industry. So most folks who have above 10 years experience, they're used to a certain way for the organizations to work. And most of the sites prior to 5 years ago, they were built for specific products in mind, and they were not flexible. So you would put $1 billion in to make a certain product, a certain tablet. So when you're coming in and trying to change the way you think about work, how you manufacture, flexibility is not ingrained. It's not trained. We don't train our folks to think that way. And the constraints are they're a myriad, just from how you do demand-driven supply, how you actually develop the products, and also how you regulate them. So we really aren't. Some folks can do a good job, but from an industry standpoint, the constraints are ingrained.
So is there a paradigm shift here on this flexibility, speed? I mean, against the backdrop of these constraints you're talking about?
Yeah, the paradigm shift is actually a kind of a twofold. About 10 years ago, when you think about there's the drug prices is an issue, and when you're also when those organizations realize that there's potential for profit reduction. What they have to do is look at, they were okay 10 years ago to have 50% capacity because they were making so much money. They can't do that now.
No.
So from a business side, it's coming in from different places, from the business side, but also from the product. So it's not just all about making one type of product that's for everybody. Now we're talking about personalized medicine as well. So when you're coming in trying to have more flexible manufacturing, because of when you have too much inventory as well, that's cash. Mm-hmm. So we don't want to. You need flexibility to reduce your inventory, but also on the product side, personalized medicine, you have to be flexible because we have with the ATMP, cell and gene therapy, that's a different ball game as well.
What are some of the key components that a firm has to be looking for to drive that to a successful endpoint?
Well, from my standpoint, and we have from manufacturing, manufacturing there. So it's. What you need to be able to do is have, it's difficult in pharma because the folks aren't brought up in it. So we need to look at other industries as well, how they look at flexible manufacturing. And before we came up here, I was talking to John about, I started off in devices, and we call it cellular manufacturing, cell manufacturing. We had to, a product took maybe a year to a year and a half to get out. And we had multiple SKUs, multiple product numbers, and we needed to be able to move our equipment around to be able to manufacture those. It's difficult for us to do that in pharma right now. So we have to be able to. The investment's different. You have to have people take chances, and with CMOs, you have to partner up with us. If you're working with CMOs, will they buy into that approach with you as well? Because they have equipment that they've already purchased that is, that's part of their capital. How do you work that?
So John, what do you guys struggle with in trying to implement flexibility?
Yeah, so just a quick overview of Adaptimmune in my background. Adaptimmune is a early stage autologous engineered T-cell company. So we have 3 products, 4 clinical trials at the moment, and we are in phase 1 in all of our trials. And so it is a one patient, one batch type of scenario. So when you talk about flexibility and speed, we think about flexibility around facility because we're early stage biotech and we're not exactly sure what's going to come out the other end. There's flexibility of operations. In the world that I live in the quick overview of how autologous T-cell therapy works, it starts from the patient's T-cells. So we first have a companion diagnostic. We test a patient for an antigen, we test them for an HLA type. They qualify for our trials. They go into the hospital. They go through a lymphodepletion process, which is collects their white blood cells. So that's our starting material. So we can't inventory starting material. It starts with the patient. From that, it's sent to a central manufacturing facility where we use a lentivirus to engineer the T cells to find the particular antigen we're going for. We grow the cells, we freeze them, and we send them back. So the operational flexibility is all around the space of the patients have to be available. Where are they coming from? They're very sick. Sometimes they miss that lymphodepletion date. So trying to manage manufacturing within that area is really challenging. And then once you do start manufacturing, it's about speed. So patients, for all intents and purposes, would like an off-the-shelf product. They would like to get their therapy right away. So the ability to quickly get through manufacturing, quickly get through QC release. One of the main challenges we're finding, to answer your question, is around batches which have deviations, which is typical in big pharma. And usually you have a big finished goods inventory sitting there, so you spend 30 days trying to sort out what went wrong and decide if you can release the batch. For us, we have to do that within hours. We have to make a disposition decision on the patient batch within hours. So there's a tremendous number of differences between there's cell and gene therapy as a whole, and then our space, autologous, which is even more challenging and it requires both. So it's not a versus, it's an and. We need flexibility and we need speed.
So why don't we expand on it a bit? So what are the unique autologous therapies that require rapid cycle times? You follow my?
Yeah, absolutely. So I think to talk about the rapid cycle time. So in particular in our space, we're dealing with 10 different tumor types, 10 different solid tumor cancer types. Phase 1 trials, patients have progressed through other therapies. So there is a need for speed to get their therapy back to them. So that's one element. The element I talked about, which is you can't start manufacturing until you have the starting material, and that starting material starts with the patient, requires the rapid speed there as well. The idea of the variability of the patient's starting material when you get into the actual process itself. So if you think about operational flexibility, now when you're in the process, how can we develop and characterize a process where your starting material is a living cell, which is going to be different if it's a head and neck cancer patient in their 70s who's had 4 rounds of chemo before this, or it's a younger person with a synovial sarcoma. All of those are different. So I mean, I could keep going, but that's kind of a list. I think there's the big operational piece, and then there's the process piece, and both of those require us to find ways to be flexible operationally and flexible to deal with variables in our starting material, and then the speed to ultimately get the therapy back to the patients.
Yeah, getting back to Mike, to me, it seems that what we consider our product knowledge and process knowledge has to be redefined in this particular situation. What do you think?
Yeah, so when it comes to, let's say, the ATMPs in general, the flexibility is needed not just from a manufacturing space, but regulatory, as well as that we call the process validation life cycle, from design and development through qualification of the product and as well as the equipment and the CPV. The one thing that I don't think most folks know is that for most of these products, versus, let's say, a typical mammalian drug product, the clinical trials are probably 10%. So they take about a couple years less time. So what you've done, and people who come in are shocked because they're like, oh, I think, because they're like, you go from they're used to, in their head, phase 1 takes this, phase 2 takes this long, and then phase 3. You're just compressing that. So what you underneath that, think about the process and product development landscape. It's compressed, and normally we have enough time to understand the attributes, the critical attributes, critical process attributes, and critical quality attributes. Many times they aren't completely 100% understood while you're going into PPQ. I'm working on some things a little later stage that we are trying to identify some of those. You may have a few on the mammalian side that you say, okay, I'm going to track and trend these, but there are a lot more because the compression that you have. So what we need to be able to do, and this is an understanding that the industry has to have is we need to spend a little bit more time on that, because when you get to that, from the manufacturing, once you get past when you validated it, if you don't have a full understanding, you're not going to get it to the patient if you don't fully understand those the manufacturing attributes. So we have to make sure that we're shifting and spending more time. So what happens is that when you're compressing everything, the organizations want to get it out, so you have this clash, this dichotomy of I need flexibility, but it takes less time, and people aren't used to that.
I think I think one of the things that you see a lot, and the analogy back to small molecule, and I know, that John, that's where John came from as well. Once we started working in areas such as oncology, we found that we had to get into the clinic fast. 90% of the time we launched with a clinical service form because we didn't have time to bring it to the final dosage form. That was almost unheard of. I mean, to the point where it became, I don't know if you were talking about a paradigm shift. It was like, what happened? And we found that, when you did the critical path, the thing that was on the critical path was not the clinical study, but the stability study. I mean, that's when I think what we have to do is we have to come to that awareness. It turns out that what we thought we had a year to do, we got 2 months. I mean, so that's it. I think one of the things I wanted to loop back to, John, is that this has got to have big impact on QC, QA, testing, deviation. I mean, how do you wrestle stuff? I mean, considering we've chopped 2 years off a development cycle, manufacturing's got to be driving nuts. And I think in the context of cell therapy, how do you deal with stuff like the QC, the QA release? I mean, how do you do that? How do you rapidly do that?
And that's another comment on autologous, which is this need for speed, right? Also early stage biotech. So typically we would outsource everything. You'd outsource all of your manufacturing, analytical testing. You might do one batch, which is going to last your clinical trial for 1.5 years. When we dose 100 patients, that's 100 batches, 100 QC tests, 100 400-page batch records. So I think you're seeing a lot of companies like ourselves trying to make a mix of external and internal resources. We've certainly, we are working with CMOs, and we will continue to do that to operationalize something that has to happen within weeks. You need to get QC results done in weeks. You need to make decisions on deviations within weeks. Bringing that in-house is something that we just recognize that we have to do. I think the supply market will mature and get to a place where they can do that, and I think there's a tremendous business opportunity for CMOs and CROs that could work in the autologous space and meet the needs of the business. But at the moment, it's a bit to your point about flexibility. They're kind of stuck in the world that, we'll send the sample there, and in 2 weeks we'll put it on test, and the test takes 2 hours, and then 2 weeks later we'll give you a report. We need it like that. So to answer your question, we're looking more at insourcing than we are at outsourcing.
I just wanted to, let's step back. You brought up supply chain. I mean, obviously, we had a discussion early in the first day here on applying the thought process to a smart supply chain. So I mean, but I think in the context of the discussion we're having, could you overview? I mean, what does your supply chain look like? I mean, I think people were a little bit stunned at how disparate the whole thing really was. Getting back, it's like what you were saying, Mike. People think it's not. It's everywhere. It looks like a fishbone diagram. So why don't we touch on what the supply chain looks like? I think that would help.
Yes.
Good?
So our space is, we talk about vein to vein as our supply chain. So from a manufacturing and supply perspective, that's from the collection of the white blood cells from the patient all the way through infusion. So our responsibility extends into the clinic, which is also a bit unique. Usually there's a handoff of inventory. Supply guys make it, we put it on the shelf, and then the operations people take it from there, whereas we're involved vein to vein. So you've got the uniqueness of the starting material, which is different. We actually cryopreserve our apheresis material, which is the starting material. We have 48 hours from the time it's collected for a patient till we can actually either freeze it or actually start manufacturing. So that supply chain adds complexity. We have clinical sites in the UK, Spain, France, Belgium. So we're looking at things like, can we get that material through customs into the States to do that manufacturing there? Or do we need to have local capability to do that cryopreservation? Brexit is causing me a heck of a lot of issues right now to understand if there's going to be a customs border there or not. So that's kind of the upfront side of the supply chain. The traditional other part of it is we use, I think, 180 or so materials on a bill of material, which are typically research grade. We're going through that exercise of trying to find GMP sources for those things. And then finally the drug product. So we freeze the drug product, cryopreserve the drug product on the outbound side. That has to make its way to the clinic, and then ultimately thawed and infused. And the other part that you'll hear in this space is chain of custody and chain of identity, right? The worst thing we could possibly do is take one patient's cells and give them to someone else. So there's a tremendous amount of investment and control around chain of identity, making sure that we know where the patient's cells have been through that whole journey, and then chain of custody, who has actually had them, and we can document that. So all of that is kind of forcing us as a phase one early stage company. We're investing in electronic batch records. We're investing in software systems to manage that whole chain of custody, chain of identity much, much earlier than you would typically see in a early stage biotech.
I mean, that's the core. It's the cell. I mean, there's got to be a lot more feeding into the supply chain. That's where, I mean, is there something else that we're missing? I mean, there's got to be. Is there other things, media?
Well, yeah, absolutely. Of course, you've got media, you've got consumables, you've got a viral vector. Let's talk about that for a moment. So for us, viral vector is the other key element. So we actually at Adaptimmune, we are also looking to internal viral vector manufacturing. That is a very, very long process. 5 or 6 months from start of manufacturing till ultimately understanding the titers and the release that you have on that. So I would say that is probably the other element that is beginning to be insourced by a lot of companies. The viral vector market, since you're using a vector for every batch, you're not setting up a working cell bank. The demand for vector is huge out there.
And that's a lot of folks don't fully understand, that part of this personalized medicine. So if you think, people can picture a tablet, and even that supply chain goes from, your API, if you have a micronization step, and then that can go to, multiple drug product locations, and they do that for a specific reason. You package locally, so from that process, it's very difficult to be really flexible, and from the supply chain, working on some of those the bulk lentivirus, the process itself is it's not the easiest thing in the world to do, to validate, and the amount of time it takes, and also some of that we brought up earlier, the QC, how to validate those things, because everything's so fast. What you have to do to completely validate the process, all those methods need to be validated. All the raw material needs to go from the clinical to manufacturing. Have you completely qualified every single vendor, and that you are shaving off a couple years, and it's frantic. And some CMOs are setting up to do the bulk, which from a flexibility standpoint, the way it's made is actually is tailor-made for, even though it takes a while, but it's not, you're in biosafety cabinets. So, and it is very manual, because it's being taken, almost directly from a research hospital into those processes. But it takes a long time, and then you're going from that bulk, and it goes into a vial, then it goes into, the drug product. That supply chain, even though the other ones talking about the tablet looks longer, this one is actually a little much more complex. You only have a couple steps, but you have to have the amount of controls you have to have, because of, you're saying the vein to vein, but also the aseptic manufacturing controls are, I would say, a little bit more. You have to have more controls on the bulk side than you do in some other processes.
I mean, I don't see any buffer here. I mean buffer as far as time buffer. Okay, let me be clear. I mean, this sounds like, it's flat out from the minute you turn it on. I mean, how do you come about that? How do you protect that? How do you protect the supply? How do you make sure the patient need is fulfilled? Because there isn't anything. If you screw up once, you're done. Literally, not literally.
That's right. I mean, we as a company, if we can't dose a patient, that stops our trial that day, right?
Right.
So that again kind of goes back to, and then there's probably a difference between an early stage biotech, like ourselves, versus big pharma who might have the resources. We have about 150, 160 people in the CMC organization for a company that's in phase 1 clinical trials, which is a big investment in an early phase trial. We talk about the ability to do second sourcing of drug product manufacturing. So for the T-cell drug product, we have one facility, we have a CMO that's also qualified, and trying to maintain a CMO, maintain harmonization of batch records, harmonization of processes. We're in phase 1 where typically you don't have to have as great a process characterization as you do, but the regulators are saying, well, you're doing it at 2 different facilities, how do you know they're the same? Usually, you're in 2 different facilities when you're in a commercial product and you have a lot more process understanding. We don't have that yet, but by the same token, we really can't afford, at least we've making that decision, we can't afford to have all of our eggs in one basket. If we have a fire in our facility, the company's done. So it's a big investment there, and it's also back to the flexibility point. I think the regulatory flexibility to understand the constraints that we have and what we're trying to do to meet the needs of a patient requires us to have this 2 facilities, early stage, lack of understanding where we're releasing just based on a C of A. So.
Well, let me just say this. I mean, and you can jump right in there, Mike, but to me, there is no difference between early and late. I mean, to me, once you get approval, you still got to make it the same way. I mean, you still got all the complexities. So, there's no, boy, good thing we got it to the commercial stage. It's easier now. Don't work that way. So can we talk about that? Well, I mean, to me, there isn't any difference.
So the one thing about these products, when they work, they work.
Yeah.
And then, so that's why most of them are breakthrough designation. So once they, they just get really, really compressed. So from an operational standpoint, for let's say coming back to the title, like flexibility, the organizations have to understand that they have to make sure that they try to do as much of that product and process knowledge characterization and capturing now. So, and sometimes it's hard because of the speed, what's happening at the same time, because they need to get to market. And what some folks may not fully embrace and realize is that from a risk management standpoint, so in ICH-Q9 it actually talks about supply. So if you can't get the product to the patient, it's a risk. And especially when that product is made specifically for them, what controls do you have to have in place for that? So when you're bringing up that the timeline is pressed, you need to be able to capture that product and process knowledge earlier to make sure that when you're in commercial manufacturing, you're not creating drug shortages, and that drug shortage is not for 100,000 people; it's for one person. So you have to be able to have those controls at that point. So it's a completely different paradigm from that. There's risk, but there's also that risk to that one patient. They don't get that product.
I think, just real quick, John, I mean, one of the things that was brought up a couple of times, and I think what brings it on home to me is it's not, when you're finally where you want to be, you scale it up. That's not the case. So that's done, right? It's a single patient, so it's always a scale out, which means you continue to clone the process. I mean, I think that's where we're going to wind up going.
Yeah, I mean, the scale out is obvious. It's one patient, one batch size. How you choose to do that, we use individual small bioreactors for the largest part of our process and the most time consuming. There's talk in this space about distributed manufacturing. Can you do this in a box that you set on the shelf at the clinic? Put your vector in one side, cells in the other, and out comes your drug product. I think we're quite a ways away from that from a quality and GMP perspective. It's an interesting place to be, and to your point also about flexibility in the industry. People who've come into the cell and gene therapy, no one's really done this before, operationalized it. There's only 2 companies that have CAR-Ts really on the market. So as we build up our team, that 140 people, most of them have come out of big pharma, most have come from biologics, small molecule background, and every day we hit something which doesn't apply. Your old way of thinking doesn't apply, and the ability to overcome those hurdles we think about it a lot. It's not so much the technical side, it's how do we actually apply what's required from a GMP perspective, and then adapt it to the needs of this unique space.
You said did you want to add something?
No, I'm just
Okay, so let me ask you this then. So how do you plan capacity utilization? I mean, why don't we both take a shot at that one?
Yeah, so the way we've been thinking about manufacturing is you think about it in terms of a manufacturing slot. So the best analogy I've used there is an airplane, seats on a plane. So we start manufacturing at this stage every Tuesday and Thursday. The patients can schedule to get on that plane. The question is, how big a plane do we make? How many seats should we have available? So we think of that as a manufacturing slot, which is enabled by trained people, it's enabled by equipment, it's enabled by having the material there. So as an example, we've kind of developed a heuristic. It's a pretty high level heuristic, but if we're going to dose 50 patients in a year, we would look at that and say, well, some of those are probably going to have to be remanufactured. You might have a fail, so we need to plan materials, let's say for 80. And then the ability to do PD work, we need to plan for manufacturing in there, maybe that's 100. So you buy materials for almost double the number of patients you think you might want to, and then the next evolution of that is a slot. So we tend to look at 50%, 40% utilization of our facility as a number to target, which is much lower than if I was in a monoclonal world, they'd be shot for that. But here, that provides the flexibility to, when patients show up, to not have a long period of time between I've screened, I'm ready for your trial, and we say, oh, you can't get on the bus until 6 weeks later. We've got to make sure that within a week or two they can get into manufacturing.
Let me ask you, I want you to talk about flexibility, but to me, when you tell a patient you can't get on the bus till 6 weeks later, and this person has been diagnosed with a pretty serious disease, that's got to generate some negative consumer feedback, I would think.
Absolutely, and this is also, just to continue to throw out things that are different, the clinical space versus commercial space for this is very different. So we're in a world now where our whole company depends on gathering data, testing patients. These patients have failed everything else. They're very sick patients. In 6 weeks, they don't have 6 weeks, certainly in certain types of cancers. Live in a world in the future where we're approved, it's working, we're a frontline therapy, patients are healthier, they can wait longer, so the need for flexibility and speed is really important in a clinical phase, but perhaps the investments you make to have flexibility and speed in the clinical space, you don't necessarily need them in the commercial space.
Right.
So it's an interesting place where we're investing in all of this capability to make sure that individual patient gets what they need and we get it quickly, which may be overinvestment for what we ultimately have in the commercial space.
I don't know, Mike. I don't see it changing a heck of a lot.
No, actually, that constraint continues once you're going, whether it's a CMO or if you're going to do some of that stuff in-house, is that you still have to do some characterization. And do you have good enough scale-up models, which are difficult, to say the least, for these type of products? So what can you do in your PD lab versus some of the additional learning that you may need for some specific mixing studies, or something you have to do to capture additional characterization work when you're right in at that PPQ and you're doing the validation.
Mm-hmm.
So it's always coming back to that understanding the product and process knowledge. Expect that. So here's the issue, is that not very many people have gone through it. So the amount of people who you can actually, go, hey, how do I do this?
Mm-hmm.
Can you help me?
Right.
It doesn't exist.
The characterization piece is another important piece of that is donor material versus patient material. And so we talk about that a lot. We do process development using healthy donor material, and we get it into the clinic. Every single patient's cells are a different phenotype and a different fitness. And so doing the process characterization, trying to figure out your process has to be flexible enough to handle that. The other part is, you mentioned small scale. So a typical phase 1 trial in this space is 30 to 40 patients. So we have a basket trial, for example, going after 10 different tumor types. So we've dosed dozens of patients in this space. I've got a couple in sarcoma, I've got a couple in head and neck, I've got a couple in urothelial. We're finding that those cells are all slightly different and the phenotypes are slightly different. So trying to, when you think about characterization, you've got the patient material that you start with, which is vastly different and unique, You've got the manufacturing process itself, which can be different, and then ultimately the clinical outcome is different. So we are just getting to the place where we're tying those three elements together, which ultimately impact our CBPs and CQAs that we have for the product and give us the ability to develop a process that can handle that wide variability but still be effective.
And it's not just the drug product. It's all, for your type. It's the bulk as well that you have to do, the bulk lentivirus process.
I look at it this way: if you know, we've all been out there, and the bottom line is, 90% of the time, if you have a problem with a product, in my experience, you can trace it back to a not deviation, but it's an excursion on a raw material who has an idiosyncratic behavior that you didn't characterize. All right, and we're talking physical chemistry. All right, now in the biologic world, and you know, a factor of 10 or 100 to that, and then match that up to the process. What I'm saying, I think that's a big mountain to climb, and it sounds like you're trying to get to it, but it's
the other manufacturing continues in the patient. Here's another unique thing that's kind of will blow your mind to think about is that. So we're delivering cells into a patient, and we measure persistence, and the idea is the cells, whether you do a vector or you do what we do, the ex vivo engineering, we expect that process to continue. So we talk about dose escalation studies, which would be traditional, and another place where you increase your dose until you find a toxic level. Well, our trials today say we will do a final dose of 1.2 billion to 15 billion transduced cells. That's the range. You really only need one of the right cells to start the process working in your body to deliver the therapy. So it's not so much a dose number, it's finding that right cell, and then having to take your process characterization all the way into the patient and consider the patient part of the factory so you understand the therapeutic effect of your product in a patient. It takes your process characterization out of the four walls of your facility and into understanding the clinical response.
So to me, we want to get to some questions from the floor, but let me ask you one last question, and that is, you know, you got to design your process to accommodate patient cell variability. How will you design a process to be flexible enough to handle the variability, which is almost counter to what we consider normal, right? I mean, you don't make your process so it can move around. You make your process so it sits still. So, I mean, how do you do that? I mean, that's got to be a challenge. Let's talk about that validation, Mike.
So, you know, what I can speak to a little bit right now is from the talking about bulk right now.
Yeah.
It's you're trying to have the windows that you have. They seem to be moving, because even right before you're doing that process validation, because you're getting feedback. Think about that supply chain. So it's the drug product process as well as the bulk. So you're having feedback loops for what's working and what isn't working. But you still, you know, we were talking about earlier the paradigm of I need to have these things because it's a requirement. You're looking at the requirement to have, how am I controlling it? What is critical? And it's difficult to get that, especially on the drug product side. There is some flexibility from a regulatory standpoint of understanding that, but you still have to be able to show that it's safe and efficacious before it goes in. The biggest area for control that you need for these processes really is on the aseptic side. That's, you know, if something happens there along the bulk, and some of these can't be filtered, and you can't, so you don't know, and it's very difficult to be able to capture that. So from the biggest area of control and that is needed is really on that aseptic side. And actually, there's going to be the Cell and Gene Therapy Conference in Long Beach in May. There's going to be a lot of discussion about that specifically. I think that's the biggest hurdle and control that's required right now for these processes.
Well, Mike, John, I want to thank you very much. Thanks.